without further ado, let's get going. So our first speaker today is Mathew Lewsey, an associate professor from Agrobio La Trobe University in Australia. So please take it away, Mathew.
Thank you, Sam. And let me find my slides. There we go. And thanks for arranging the time travel so that I can speak to you from Friday morning. So I'm going to talk to you about gene regulatory dynamics in germinating seeds. And I'm going to talk about the work we've done using bulk tissue and how this tells us a lot about seed germination, but there are limitations in this. So we've moved towards single cell resolution. And in the spirit of this being a community meeting, I'm going to show you pretty much everything's unpublished apart from bulk tissue work. And I'm going to point out where we found challenges along the way that hopefully will help other people improve their experiments. So my lab is very interested in how seed germination occurs and particularly how the genome is regulated as this occurs. The reason this is interesting is because an awful lot has to happen in a very short period of time. So a seed, this happens to be an Arabidopsis seed, will go through a process of germination in a short period of time, maybe 3-4 days, depending upon the conditions. And it's going to start from the dry seed that you see on the left there, where the genome's all packed down, it's heterochromatic, it's dry, it's metabolically inactive. Upon perceiving the correct conditions, it's going to imbibe water rapidly. In a matter of minutes, respiration will go to a very, very high level, gene expression will start, translation will start, and the cells will start to grow by expansion. It will then proceed through what's termed tester rupture. This is where the endosperm ruptures out through the seed coat, and then germination in the strictest sense ends as the radical or the nascent root emerges through the seed coat. It's very important that this is correctly controlled, is in tune with environmental conditions, because if the seed picks the wrong time to germinate or fails to choose to germinate, then the plant doesn't grow and no further seeds are produced, so there's no reproduction. The plant has effectively failed. And what we started out trying to understand was how much gene expression changes across this period of time and how that's controlled. In order to do that, we conducted bulk transcriptome profiling across a time series of germination of Arabidopsis seeds, as you see here. We went from the dry seed through the period of stratification, then through the transition into the light and effectively room temperature to the end of germination. And we took about 10 time points, profiling the transcriptomes at each stage. And actually in concert, we looked at the DNA methylomes and the small RNAs as they change. So we first asked, how much does gene expression change? And the answer was an awful lot. So what I'm showing you on here is a heat map of all of the different genes or transcripts that are changing the expression over germination, and time is moving down on that heat map. So the dry seed is at the top, the germinated seed is at the bottom. It's getting pretty close to a seedling at that point. And we found that 24,283 transcripts were differentially regulated across this period of time, out of about 27,000 genes in Arabidopsis approximately. These fell into interesting clusters of regulation. So you see in the center here, find the pointer, this one. You see in the center here, there's a transient upregulated cluster. So these guys go up around about the transition between stratification into room temperatures and light. You see a late upregulated cluster over here. So this is really kind of correlating with the developmental transition or the metabolic transition into harvesting light. So you're going to have certain machinery associated with that. And then there was this quite interesting cluster over here of downregulated transcripts. So these guys had apparently been packed very, very highly into the dry seed and stored stably, presumably to be translated and used very early in germination, and then the transcripts have degraded away over time. And in concert with this, we see a broad loss of DNA methylation throughout the genome. So DNA methylation seems to be going away as the genome unpackages and becomes expressed. And we see large changes, dynamic changes correlating with the transitions in small RNAs. So the small RNAs are changing in concert with DNA methylation, and also as we transition from dark into light, for example. We could use this data to build quite nice transcriptional models from which we could predict regulatory transcription factors, which we could pull out, mutate, and we found they actually did interrupt both the rate of germination and the gene expression program during germination. And we did that all from bulk transcriptome data. So we were pretty satisfied that we could use that. But everybody in this call at this point is probably aware that there's also a big simplification in here. A seed is actually a mixture of very many cell types, and that's all been mixed up in this experiment. We've got useful information now, but we've lost a great deal of detail. That's how I change my slides. So here's a picture of an Arabidopsis seed. It's sectioned through, and we've labeled on the major organs and tissues. You've got the cotyledons at the top, you've got the hypocotyl down the bottom here, going into the radicle. All of these organs are composed of different cell types. You can see the provasculature through the middle that's going to become the vasculature, and up in the cotyledons here, and individual constituent cell types, which contribute to the function of those organs and individually to the success of the plant. Now, not only are we mashing up in our bulk experiments all these different cell types, but these cell types are doing different things at different times. There's some lovely work, for example, from George Bassel's lab, which shows that there's very tight spatial localization of hormone perception in a germinating seed, and that growth rates of cells vary within individual organs. So different cells within the hypocotyl and radicle, for example, are growing at different rates at different times. So if we wish to really understand how a seed is regulating its genome during germination, we're going to need to go to single cell resolution, and that is what we did next. We designed an experiment that looks like this. Basically, we took 3 time points. We took 12, 24, and 48 hours, which is effectively early, mid, and late germination. This is during the light phase of germination. And we decided to simplify the experiment somewhat for a couple of reasons. By releasing the embryos from the seed coats and enriching them. So we've popped open the seed coats, we've pushed out the embryos, and we've discarded the seed coat and the little bit of endosperm that you find surrounding the embryo. We saved some of those bulk embryos, and I'll come back to why in a moment. And then we isolated protoplasts from what remained. And so at this point, we've got protoplasts with which we can do single cell RNA-seq. We took that through to the 10x platform. We need those for our microfluidic approach. But the first thing we did was actually a comparison by bulk RNA-seq of protoplastid cells versus non-protoplastid cells in order to understand the effect of protoplast isolation on germinating seeds. The reason we did this was because we weren't certain if the available lists of protoplast responsive genes from the literature would really tie through well to our particular experimental model here, our experimental stage, because they come from quite a different stage. So the first thing we did was look at how many genes and which genes were affected by this. So we compared by bulk RNA-seq, we compared our protoplasts with our non-protoplasts from the germinating embryos. What we found was, in total, something like 2,000 transcripts are responding to protoplast isolation at this stage. Now, previous studies, some of the published studies on roots, for example, have used a nice data set from Ken Birnbaum's lab, which defined protoplast responsive genes. When we compare our particular growth stage to that list, we actually find that, looking just at the upregulated genes, we've got about 1,400 upregulated genes, and only about 109 of those are shared with the list from the Birnbaum experiment. So that particular data set is not useful in our growth stage. So that was a useful experiment for us to do. This informs us that we need to understand which genes are responding to protoplast isolation. They might well be stage specific. Conveniently, what we also found, though, was that these are not time point dependence. So we've looked at 12, 24, and 48 hours. The same genes are responding to protoblast isolation at each time point. It seems like there's a protoblast gene set for seeds. So now, having figured out that we need to gray list this set of, in total, about 2,000 genes up and down regulated, we can move on to looking at our single-cell RNA-seq experiment. So that was designed and analyzed like this. We've got germinating seeds at 12, 24, 48 hours. We've done this in biological duplicate, and this yields about 13,000 cells sequenced in high-quality cells sequenced in our experiment. We're then going to integrate the data from the replicates and the different time points, and we're going to identify clusters of similar cells. And up to that point, it's relatively straightforward. Methods exist to do this. The last thing we're going to do is annotate cell identities, and this is where it became more challenging. The particular challenge being that there's not a nice kind of defined map to ground truth our single-cell RNA-seq. So in the root studies, for example, there's the great work that came out of Phil Benfey's lab, where they had painstakingly isolated by FACS all the different cell profiles of the roots. And so there's this ground truth you can go to and say, this is the transcriptional signature of each cell type. In our case, that hasn't been done. And so what we have done instead is the postdocs working on this project have combed through the literature very carefully and looked at many different papers that have analyzed different cell types of germinating seeds to identify marker transcripts that identify those cell types. And then when we have our clusters of germinating seed single cells, we can map on those marker transcripts and we can say, okay, this is this cell type, that is that cell type. And this took quite some time. This is a slow process, but ultimately we were quite pleased with the outcome. And so we ended up with something that looks like this. This T-SNE plot shows all of our cells from all of our replicates and all of our time points, so 12, 24, 48 hours. And we were able to annotate pretty much all but 2 of the 15 clusters on this plot. We can annotate them broadly to organs and actually, in most cases, annotate them down to cell types as well. And so what we found was a nice cluster of cotyledon-specific cells. We found a nice cluster of the provasculature cells, several clusters relating to hypocotyl, and then interestingly, some clusters that come from the radicle, which you can see overlap with some of the hypocotyl clusters. So you have some radical-specific clusters there, hypocotyl-specific over there, and then a set that are shared. Now, if you picture or remember the image I showed you of the section seed earlier on, the hypocotyl and radical kind of merge together, and it's not necessarily unreasonable to expect that somewhere in a boundary zone, there are going to be cells that appear somewhat shared. They have signatures of both organs. And lastly, we had a couple of unknown clusters, which we have some ideas what they might be. We think this might be to do with the cells turning on very early during germination and not having clear markers to define them by. So we did all this through literature creation effectively, but we wanted to go back and check that our annotation really was accurate, that it was borne out in the seed as it germinates. And so we selected clusters, and from those identified new marker genes that defined the cluster that were computationally identifiable in the cluster, but that we had not used previously to annotate those clusters. So these are independent markers. And we went back around and checked those by RNA in situ to see, okay, where is this cluster and when is it, and does that fit with our annotation? The results were that actually it fitted quite nicely with our annotation. On here, I'm showing you 2 different clusters that we've checked. One is cluster 9, which was annotated into the provasculature in the xylem, and one is cluster 13, annotated into the cotyledons in the mesophyll. You can see here for cluster 9, provasculature xylem, there's this nice tight signal in the provasculature, and actually if you look carefully, you can see it through the center here as well. And similarly, for cluster 13, you can see this nice strong signal that's in the cotyledons. And so that was very satisfying. Our hand annotation has worked and it's borne out in reality in Planter. This experiment, though, it highlighted something important about the data, both of these clusters were only detected at 48 hours in our single cell experiment. And when we do the RNA in situ with the marker genes, we only see them present at 48 hours. At a 24-hour RNA in situ, you see there's no signal from either. So this suggested that there was some transition in the clusters over time, that there's something changing about the way we detect clusters from 12, 24 to 48 hours. And so we examined that next. On here, I'm showing you the same t-SNE plot from earlier with all of the cells from all of the time points, but instead of labeling by cluster, I've labeled them by the time point at which those cells came from. Here you see that there's a distinct region of 12-hour cells. That's neighboring a region of 24-hour cells, but they're not the same. They're not clustering directly together. And then you've got a very large patch of 48-hour cells over here. So they're all separated on our plot. This indicates that they have different transcriptional signatures. In some situations, you might imagine that these are, for example, cells dividing and differentiating down different paths, but that can't happen at this growth stage. There's no cell division and no cell death during germination. All the growth in a germinating embryo is occurring by cell expansion. So instead, this suggests to us that we must have the same cells present at each time point, but they're transitioning through different transcriptional states. We looked at that in detail next, basically by assessing which clusters do we detect at which time points. I'm showing you that on this plot. So on this plot, we have each of our time points, 12, 24, 48 hours. Clusters are all in different colors. And at each time point, I'm telling you the proportion of the sequence cells that was annotated to a cluster. Looking at this very crudely, you can see the colors change. And that indicates the clusters we're detecting change. So we have different clusters that we're detecting. And actually, if we look, we're detecting clusters from different tissues and cell types over time. If we focus in just on the hypocotyl, and particularly I've labeled hypocotyl cortex clusters, so all the same cell type, 1, 5, and 7. At 12 hours, we detect a little bit of cluster 1, a lot of cluster 5, and basically none of cluster 7. As we go through to 24 hours, we've now got a lot more cells annotated to cluster 1. We've lost some of cluster 5, and cluster 7 has greatly enlarged. By 48 hours, we're barely detecting these. So again, this highlights, or this suggests that we're seeing, since we have no cell division, no cell death, we're seeing the same cells at different time points transitioning through different transcriptional states. That is what this implies. For that to occur, we would expect there must be high levels of transcriptional activity to reprogram the cells. We examined whether that was the case, or whether that might be the case, using a metric called RNA velocity. So RNA velocity is basically an assessment of how highly active transcription is, and it also gives you some indication of the trajectory that a cell is moving along. We focused in just on the hypocotyl, so all of the clusters annotated to the hypocotyl, but particularly on these 3 clusters, 1, 5, 7, that were all hypocotyl cortex cells. And if I show you that data, so they're presented on UMAP plots on here. So on the left-hand side, I've just shown you the hypocotyl cluster identities. This big blob shows you clusters 1, 5, 7, all hypocotyl cortex cells that are grouping together. You can see we've got some of 5 up here, and it kind of spreads a bit down toward the bottom. We have 7 is going the whole distance of the cluster or of the group. And then we've got 1 down the bottom here. Now, if I take the same plot but change the labels to the time at which we detected the cells in those clusters, if we look at our clusters 1, 5, 7 hypocotyl cortex group here, we seem to have 12-hour cells at the top, a lot of 24-hour cells across, and then a small number of 48-hour cells at the end. Next, we're going to look at the RNA velocity and say, okay, do we have high RNA velocity? So a lot of active transcription that might suggest some way in which we can get from 12-hour cells through to 48-hour cells. And that's shown over here. So the intense yellow indicates that, yes, we do detect high RNA velocity. So there's an awful lot of active transcription in this group of 3 different clusters. And the arrows give some indication of the trajectory that the cells are moving along, the direction they're heading. And you can see these arrows generally trend from the 12-hour down toward the 48-hour, which would bridge across the 24-hour. And if we do proper trajectory reconstruction, we can actually find a path that moves down through these 3 clusters toward this state. So we think that these 3 different clusters represent the same cells at different time points moving along a transcriptional trajectory. And the next thing that we need to do with this is start to understand, okay, how might that be controlled? So can we start to pull out the transcription factors that control movement of transcription along this path? With that, I'll conclude. So I've told you that substantial remodeling of gene expression occurs during germination. Seeds are very complex structures, and these are composed of many cell types. And this limits what we can find from bulk analyses now that we have single cell techniques available. Using the single cell methods, we can better characterize which cell types are active where and when during germination, and that's what we've done here. The only thing that remains for me to do is to thank the people who did the work. So the 3 main people driving this are Lewsey Liu, who's done essentially all of the lab work on this and the beautiful RNA in situs and things like that. And then Quentin Gould, who used to be a postdoc in my lab and now is at Weihai, and a student of his, UAU. They've been doing all of the analytics. We've had some awesome collaborators, particularly from Jim Whelan's lab, from Matt Ritchie and Marnie Blewett at Weihai, and from Ryan Lister's lab at the University of Western Australia. And with that, thank you for your attention. I will gladly take questions.
Okay, wonderful. Thank you for that excellent talk. So we have a question from Thomas. Very interesting talk. From the 24,000 differentially expressed genes in the bulk data, about 1/3 seem to be downregulated over time. Does that mean that some more than 8,000 genes are actively being transcribed in dry seeds?
So what we think is happening there is that these are actually being transcribed during seed or during embryo development, because this is a very active part of development. And so those transcripts are going to be transcribed then, packed down and stored very carefully. And the remarkable thing is that they're then stable when a seed is dry for 1, 2, 3, 4, 5 months, right? That they're still there. But then presumably they're going to be, the transcript's going to be there, ready to go to be translated, and those enzymes or proteins can be used very early in germination.
We have a question that was originally in the Zoom Q&A. So as a reminder, please put your questions in the chat from the Cell Events platform. So did you also analyze recalcitrant seeds?
Recalcitrant being, I'm not
maybe
exactly
non-germinating,
sure
I wonder.
Oh, no, recalcitrant seeds are seeds that don't really get, they're desiccation sensitive. So they can't get dried.
Ah, okay.
Orthodox
right.
seeds are seeds that are desiccation tolerant.
So we did not analyze those. I think that would be extremely interesting though.
Great. So we have a question from Alex. Have you checked the identity of unknown clusters since they're highly represented in the 12-hour but not other time point data sets?
Yeah, we just started looking at that. And again, this comes to this limitation of the extent to which you can find ground truth data to try and figure out what things are. But it looks like possibly this is a general state of the cells just at the very start of germination. That's what we think so far. I'm really telling you, like, this was a discussion I was having 2 days ago, so we haven't validated that yet, but it seems like maybe the cells have a more general state and then they start to germinate and they become more specified into what they're doing as germination proceeds, possibly.
So we have a question from Maurice. The unknown clusters 8 and 11 in the first 12 hours might have something to do with programmed cell death because protoplasting brings a lot of stress. So I guess that's more of a comment. Kaisa asks, what do you think your RNA velocity findings say about the way we interpret single cell data regarding cell identities?
I think I guess the main thing I'm trying to get across with the work I've just presented is that in these systems where we have the same cells and they're not dividing, they're not changing their identity, so to speak, but they are changing potentially their function. And so they're actually remodeling transcription in quite a large way. And so when we start to do cluster identities, it's unlike a mammalian system where they're trying to cluster out individual cell types. That's not really what we're doing. We're trying to understand how cells transition along their job as they exist. I think the RNA velocity is a useful tool for us to start to get a handle on the direction the cells are going, the fate they might adopt as they move along that path. And it tells us, that we have very high transcription in these cells that seem to be changing. But we definitely need to, and we are doing full kind of trajectory reconstruction to start to see if there is, or we have found that there is a path that moves between these different cell states that was indicated by the velocity analysis.
And so we have two questions from Pavel. The first question, very nice result. How did you exclude the hypothesis that cells from the different time points cluster into separate clusters as a result of developmental stages rather than a consequence of batch?
Yeah, so I would have to defer to my postdocs for the precise details of how they analyzed it. But basically, we've got a couple of biological replicates in here, and we're looking for reproducibility between replicates. Now, we certainly find some variability between independent biological replicates, and these were truly independent, done at completely separate times. We see there is some differential recovery of cells from each cluster type. But broadly speaking, when we do independent replicates, we do find the same cluster, the same distribution of clusters across time points. And we see this general trend across replicates of one time point being very rich in certain clusters, the next time point being rich in different clusters, and so forth.
Okay, and then the final question is, do you have any idea how big the proportion of non-spliced transcripts are in your data? And then we'll have to move on to the next speaker.
I'll give you a short answer off the top of my head. I do not know the proportion, but the RNA velocity analysis shows that we've got an awful lot of the unspliced transcripts, because this is how we detect active transcription in those particular cell types we were looking at.
Wonderful. Okay, and then there's at least one additional question for you, Mathew. So if you don't mind answering that question in the platform, we will move on to our next speaker.